So I'm really excited to be able to host Ellie Rando for the genetic seminar today. And I'm sorry that we can't host you in person, which would be a lot more fun all around, I'm sure, but at least we have this as a substitute. So Ellie got his BA from Harvard and worked with Tom Maniatis, so started out with kind of a star-studded pedigree there. And then went on to do an MD and PhD at Stanford, where he worked with Jerry Crabtree. And from there, in a move that I'm sympathetic to, decided not to do more clinical work, but went on to be a Bauer Fellow at Harvard. So he started his own lab right away, and did a bunch of pretty amazing work on yeast chromatin biology. So including making every possible combination of lysine mutations on histone H4 to test the sort of function of the histone code, and a whole variety of other really exciting chromatin work. So when I started in this field, I would go to meetings and basically every talk would reference Ollie's work in some way or another. So I was like, I have to meet this person at some point. So he then moved to UMass Worcester, started as a junior faculty there, and continued the yeast work, but also made the move into mouse, where he started looking at epigenetic inheritance across generations. And he had very quickly a very foundational paper in the transgenerational epigenetic inheritance field, showing that paternal diet affects offspring phenotype. And that's sort of been the basis of a ton of studies since then in his lab and elsewhere. And what he's done really nicely is to take this phenomenon and really pick apart a mechanism for it. It's a really tough thing to do. And hopefully you'll hear some of that today. It turns out that this is mediated, at least in part, by small RNAs that are coming from somatic tissue, which is really interesting. And one of the things that's been just sort of really nice I always really like about reading or seeing Ollie's work is that it's just very clear-eyed about the data. He has really, really bold ideas and finds ways to make them testable. And then goes where the data goes, which is great. So it's never, pursuing a premeditated hypothesis, really. It's figuring out what the data is telling you, and really understanding the system, which is really inspiring to me as a scientist. I'm sorry that he can't be here in person because he gives a really just amazing talk in person. I'm sure it'll be an amazing talk via Zoom as well. So with that, I'll hand it over to him. Excited to see what he has to say.
Okay, well, thank you, Dr. Lesh, for the very kind introduction. Yeah, I'm sorry I can't be there in person either. I sort of have a family history with Yale. My father got his PhD from Yale in chemistry in the 1960s. Got his PhD, it took him 2.5 years, and he spent 9 months of that in a full body cast from a motorcycle accident. So I sort of started out with kind of unrealistic expectations about how long graduate school would take. Anyway, I haven't been back to New Haven since I looked at it for college 25 years ago. So anyway, I'm kind of bummed I can't come hang out with you guys. Anyway, hopefully I'll make your Zoom time worthwhile here. As BB said, my lab has generally been interested in epigenetics from the beginning. Our work in yeast was sort of aimed towards trying to understand how you replicate chromatin states, although we've sort of let that go by the wayside in recent years. So just to define the term, since the term epigenetic means so many things to so many people, there are at least 3 different uses of the word, starting with Waddington. I guess it started with Aristotle, but in modern biology, starting with Waddington in the early 20th century. The definition that I prefer is the one that means inheritance. So a trait is epigenetically heritable. This is the Riggs and Holiday definition from the 70s. A trait is epigenetically heritable if two organisms have the exact same genome but differ in some phenotype, and can pass on that phenotype without a change in DNA sequence. And the classic version of this is in a multicellular organism. All your cells have the same genome to a first approximation. When a liver cell divides, it never makes a skin cell or a kidney cell, it makes two liver cells. So you pass on both your genome, the book of instructions, as well as state liverness, and state liverness is epigenetic. Now, of course, this is pretty well understood. People have studied cell fate inheritance for many decades and have defined three major epigenetic information carriers. These are DNA modification, small RNAs, and chromatin packaging. Now, that's cell state inheritance, and of course, in a multicellular organism, this is not the way we divide. In order to pass information from one generation to the next, intergenerational epigenetics, the epigenetic information has to be loaded into these very specialized cells, the gametes, the sperm and egg. And then at the spark of life, at fertilization, all of the epigenetic information which has been transmitted by the sperm and the egg has to then unfold over the course of development to give you a coherent physiological phenotype. Now, I wouldn't be telling you this if there were not evidence that epigenetic information does make it through the germline. To my understanding, the first discovery of this was with the phenomenon known as paramutation. It was originally discovered by Bateson and Pellew in the rogue garden pea in the 1920s. It was then given a name and studied in more detail by Brinks and Co. in the 1950s with maize. And so I'll briefly tell you about how this looks. So you can have maize as corn, you can have purple or green corn. They differ in the expression of a transcription factor called B1, which drives the anthocyanin biosynthesis pathway genes. So purple corn expresses B1, green corn represses it. If you cross these to each other, you will get green offspring. And now, if this were a Mendelian trait, this would mean that green was big B, purple was little b, and these would be heterozygotes. But now, if you take these green offspring and back cross them to purple, if this were a Mendelian trait, you would expect half purple corn and half green corn. And what you get is completely green corn. And this can persist through hundreds of generations of back crossing to the purple corn. So it almost seems as though the repressed allele here is infecting or paramutating the active allele, hence the name paramutation. And so decades of genetic study of paramutation and similar types of phenomenon, with paramutation largely by Vicki Chandler, have defined the three major epigenetic information carriers: small RNA pathways, DNA modification, and chromatin of this silencing phenomenon. So some epigenetic information, as you can see here, does get passed from one generation to the next. Now, the fact that some epigenetic information does make it into the germline sort of has resurrected an old idea, which is that unlike DNA sequence, which is largely unaffected by environmental conditions, epigenetic marks are very sensitive to the conditions a cell is growing in. So if you heat shock budding yeast, the chromatin state of thousands of nucleosomes changes. And so because of that, you can imagine that the world a parent experiences could transmit information or affect the physiology of future generations. This is often called the inheritance of acquired characters or Lamarckian inheritance, although that's a misnomer for many reasons. And of course, it was a very popular idea in the 19th century. It was generally considered to be totally discredited by August Weismann in the late 19th century. So this is a German embryologist. He defined the soma and germline distinction, right? So the immortal germline is what links us to our ancient ancestors through millions of years of inheritance. The soma is the meat machine that carries around the germline looking for other germlines. And so what Weismann did is he cut the tails off of mice and he mated them to each other and he measured tail length and offspring. He sees no difference in the tail length in the next generation. which he used to refute the idea of the inheritance of acquired characters. There are a couple of problems with that experiment from a modern point of view. Maybe the useful thing to do if your parents tell you that someone's going around cutting tails off is not to have a shorter tail, but is to run away from people in lab coats. Even at the time, it was a little bit of a rigged experiment. Well, not rigged, but not a perfect experiment in the intellectual context of the time. Even Lamarck recognized that soldiers from wars throughout history who've lost their arms don't have kids with short arms or lacking arms. So even in Lamarck's formulation, there had to be some element of utility or desire to pass on environmental changes. So a mutilation was maybe not the best system for this. But in any case, this was a really influential refutation of the idea. And that really persisted until the beginnings of the discovery of epigenetic phenomenon sort of brought this idea back into at least speculative discussion. And so that sort of brings us up to when we got involved. And so we set out to try to figure out whether or not ancestral environmental conditions would affect phenotypes in future generations. So we do this in inbred mice. So hopefully we're not like selecting on sperm with an A at nucleotide 1000 versus a G, like you would have in humans or something. We do this as a paternal effect because in mammals, mom is baby's first environment. So fetal alcohol syndrome is not magic in epigenetic, you're just poisoning a fetus. Whereas with dads, we can set it up so that they just leave sperm in a memory. So what we do is we split male litter mates to control, and we have studied low protein diet the most, although we now have looked at a number of different conditions, including high fat, nicotine, I hope to be able to tell you about at the end, so on and so forth. We raise them to sexual maturity. We mate them to control females for 1 or 2 days. We then take the males out, so hopefully they're not directly, the kids are not directly interacting with the fathers. We're trying to minimize things like microbiome transfer through defecation in the cage, so on and so forth. We then let the pregnant females have kids. At 3 weeks of age, we grind up the kids and we look at gene expression by, back then it was microarrays. And so basically there are 2 major differences between the experiment we did and the Weissman experiment. Both of them, you're poking an animal and looking at its kids, but we're choosing a stimulus that it makes sense to tell your kids about. And if people want to ask about that in questions, what I mean by makes sense, I can get into it. And the other is that instead of looking at 1 quantitative trait, which is tail length, we're looking at 30,000. So it turns out hundreds of genes change expression in liver depending on what dad ate. Here's a typical microarray heat map where every column is a comparison between a low protein animal, low protein offspring and a control offspring. Red means that the gene was upregulated if dad ate a low protein diet. Green means it's down. And so you can see that there are very significant, albeit not completely penetrant, gene expression changes in the next generation. The downregulated genes, the upregulated genes are very heavily enriched for genes involved in cholesterol and lipid biosynthesis. This is more than just a gene expression phenotype because when we do metabolomics in the liver, we see significant decreases in free cholesterol and cholesterol esters. Now, this is all I'm going to say about the phenotype in the next generation. I don't know the utility of this for a low protein future, let's say. People who know about metabolism say that a stronger phenotype for cholesterol esters over free cholesterol suggests a bile acid phenotype. So perhaps the low protein kids are dumping more bile acid and they're homeostatically upregulating cholesterol biosynthesis to keep up with the loss of cholesterol. That's as far as we go here. This turns out to be redolent of phenomena that occur in humans as well. Oh, sorry, this is just the other systems. Okay, so this is our contribution to this field. Contemporaneously and since then, there have been, at this point, I would say hundreds of papers that have linked different paternal environments to changes in offspring phenotype. So here are all of the metabolic or dietary ones. So This is actually the paper that pushed us out the door. So in rats, dads on high-fat diet have kids with glucose control issues. This is basically our system a couple of years later, where in addition to the cholesterol phenotype, they see like blood pressure changes and again, glucose control. All of these systems are fairly interesting because what they're doing is they're starving a pregnant mom and then taking her son, who was in utero during the starvation, either starving or giving them high fat, and then using that son as the paternal generation. And so these also affect lipid handling and glucose control. But it's quite interesting because the reason people chose this type of stimulus is that fetal development is when primordial germ cells develop and when cytosine methylation patterns are established. And so I think it'll be really interesting to compare the post-birth and the pre-birth stimuli in terms of what kinds of phenotypes they induce in the next generation. Beyond dietary phenotypes or stimuli, there are cold and exercise affect metabolism in the next generation. Stress affects stress responsiveness in the next generation. Small molecules and toxins ranging from cocaine, alcohol, nicotine, and others affect the next generation as well. So again, at this point, I would say there are several hundred papers that have linked a male's environmental conditions to physiology in the next generation. This also looks like something that people see in humans. So the most famous example is the so-called Dutch hunger winter. So this is at the end of World War II in response to a transit workers' strike. The Nazis starved the Dutch people. They burned all the food stores and salted the fields. Government rations got down to 700 calories a day. People were eating tulip bulbs to survive. Something on the order of 20,000 people starved to death. This lasted until the Allies restored food control in the spring of 1945. And so what's rare about this, there are many famines in history, but it's rare for a first world country to go into a famine and come out with defined kinetics. And it turns out that children who were in utero during the Dutch hunger winter have elevated rates of diabetes, obesity, and cardiovascular disease, glucose handling and cholesterol handling phenotypes. Now, this is quite interesting because this is kids starving in utero, so this is a maternal effect. But you see the same kinds of phenotypes as you see in paternal effect systems. And I think that might be a really interesting hint to how these things work. In addition to these female line effects, there is a little evidence in humans for male line effects. This comes from the so-called Uvercalyx cohort. It's a remote parish in Sweden where they started keeping really good crop records in the 19th century. And what Kati, Pembry, Bygren, and others have done is link modern morbidity and mortality to ancestral food supply. And again, they see that your paternal grandfather's access to food is correlated with diabetes and obesity in your grandson. So people have a lot of issues with this particular study, but there's at least some hint that there are male line effects of nutritional status in humans as well. So our interest, as we come from the world of molecular mechanists, has been to try to understand how these systems work. So to restate what we're trying to understand, you put males on different conditions, mate them to access a wide range of altered physiology in the next generation. The assumption is that this is going to be transmitted through epigenetic marks in sperm. It's worth pointing out there are other ways that fathers can talk to their kids. This includes seminal fluid. There's a really robust literature, in particular in Drosophila, from Mariana Wolfner, showing that seminal fluid components can alter offspring phenotype. Cryptic maternal effects, this is largely literature in birds, Goldian finches in particular, but females can judge mate quality and allocate resources accordingly. And so in principle, a control female could be judging our low protein males and restricting blood flow to the placenta. And then we don't have it on this cartoon, but the microbiome can also be transferred and affect kids. So the first question we have is the information here in sperm? So to get at this, we purify sperm and eggs from males on different conditions and control females. We carry out in vitro fertilization. We transfer those embryos to a second female, and we've been able to reproduce our cholesterol phenotype through IVF. So that tells us what you would have expected, which is that the information is in sperm or co-purifies with sperm. This is actually quite nice for many reasons. One of them is that we've started to take maternal effect seriously now that we are splitting females into the oocyte donor and the uterus donor. And so that's something that's just sort of beginning in our lab, but we're quite interested in oocyte epigenetics now as well. Now, as far as now that we know the information is transmitted in sperm, of course, we're going to be looking at all of the putative epigenetic information carriers in sperm. I don't really have time to get into it, but I'd say we're quite sure that in our case, the answer is not cytosine methylation. I'm skeptical that it's going to be histone retention in sperm, although I'd say less confidently that we've ruled it out. And so we're going to be focusing on small RNAs. I don't think we have shown that small RNAs are the carriers here. What I will tell you is that I think everything we've done to look at small RNAs in sperm has led us into really interesting new areas. We've made a lot of interesting observations with sperm small RNAs. Our interpretation of some of those observations I'm kind of curious about now. Okay, so small RNAs are central to the really well-studied transgenerational epigenetic systems, paramutation maize, RNAi in C. elegans. So we've started to pay attention to small RNAs in mammalian sperm. So mouse sperm has a really unusual payload of small RNAs. So this is just deep sequencing of 18 to 40 nucleotide RNAs. This is the size distribution of the inserts. So here at 21 nucleotides, you have your microRNAs. In a fibroblast or hepatocyte, that would be the vast majority of what you clone. And we see about 10% of small RNAs in sperm are microRNAs. But the elephant in the room here is this pretty unusual peak from 28 to 35 nucleotides, all of which are derived from the 5' ends of mature tRNAs. So those are called tRNA fragments or TERFs. The most abundant small RNA in sperm is TERF glue CTC. It's about 20% of all clonable small RNAs. Then we have VALCAC and TERF glide GCC, each at about 5%. I'll tell you now, and we'll come back to it later. We now are we've always wondered where the 3' ends of these tRNAs are. And it turns out sperm are filled with the 3' ends as well, which very much changes how you think about what sperm are carrying. They simply aren't clonable using the Illumina TrueSeq protocol, which is what we used in this case. But if you mess around with your cloning protocol, you can actually access the 3' ends as well. Anyway, when we compare control and low protein sperm, what we find is that low protein sperm carry about 2 to 3-fold higher levels of TERF glide GCC. So I'll be calling it TERF GG for short. So this caught our attention for a bunch of reasons. First is that, of course, you would tell your kids about a low protein diet using small RNAs made from tRNAs. Small RNAs are how parents talk to kids. tRNAs very obviously see metabolism fairly directly. Also, if we think that the RNAs in sperm are going to do anything in a zygote, you really need it to be the most abundant RNAs in sperm, because sperm are 1,000-fold smaller than an oocyte, and I believe they have less RNA on a per femtoliter basis. And since this is the third most abundant RNA in sperm, this sort of tracks. And then the last reason, but it turns out tRNAs are universally used in LTR element replication. So this is true for retroviruses like HIV, which is packaged in the virion with a tRNA bound to what's called the primer binding sequence. So when HIV infects a cell, reverse transcriptase primes off the tRNA to make one long terminal repeat, then you make the cDNA to the whole thing. And this is true of both retroviruses, but also endogenous retroelements. And so you can imagine people talk about piRNAs as a genomic adaptive immune system. tRNA fragments could act as a genomic innate immune system. By clipping your own tRNAs, you could fight off an LTR element even if your genome's never seen it before. And because epigenetics tends to be about control of selfish elements first, that then gets co-opted for other forms of regulation, this got us sort of to pay attention to tRNA fragments. So I'm going to tell you a little bit about the biogenesis of tRNA fragments and a little bit about their function. So the dogma about RNA contents in sperm is that they are the degraded remnants of the RNAs you use to make a sperm. So you go through the process of spermatogenesis, you have a bunch of RNAs that are involved in the process, and then you chew them up and you just have these sort of trashy old remnants of RNAs that sperm continue to hold on to. So the null hypothesis here would be that if we look at the testis of a low protein animal, we have higher levels of tRNA glide GCC. So as we started to chase that down, we started to realize that these tRNA fragments don't even seem to be made during the process of spermatogenesis. So to tell you about this, I have to introduce you to the mammalian reproductive tract. So the testis is the site of spermatogenesis. So from spermatogonia through meiosis, through spermiogenesis, you make morphologically mature sperm at this point, which are incapable of fertilization. So they don't know how to swim yet, and they don't carry a lot of the proteins they need to recognize the oocyte. Sperm then spend about 2 weeks going through a long convoluted tube called the epididymis. They proceed from caput to corpus to cauda. This is the storage organ. Ejaculation is a muscle contraction around the cauda, and you squeeze sperm out the vas deferens. Here's the original description of the epididymis by Renier de Graaf in the 17th century. And you can see how this tube is all squiggled up. So in humans, the tube is about 2 meters long, and sperm spend about 2 weeks going through it. The epididymis is the least studied organ in the body, probably because it doesn't get cancer, which is interesting in and of itself. My favorite factlet about the epididymis is that in a 2005 German newspaper poll, it came in third among people's favorite male reproductive organs, which means that we beat the prostate. So anyway, it's kind of mysterious what the epididymis does. So here's an image of a mouse epididymis by light sheet microscopy. You can see how you fit about 1 meter of tube into this little nugget of tissue. And again, sperm spend like 10 days in here swishing back and forth. Each of these segments has a completely different luminal microenvironment. So if you just look at gene regulation across the epididymis, there are genes that are expressed at 100-fold higher levels here than here in the caput than in the cauda. A lot of the things we know about the epididymis in terms of what it does to sperm are things like bicarbonate manipulation or calcium signaling. And these are things that take like milliseconds, and yet sperm are spending over a week going through this tube. And so what we believe is happening in the epididymis is that we're pretty sure that some of the RNAs that sperm deliver to the embryo are actually first synthesized in this tissue and then trafficked over to developing sperm. I'd say we're 95% sure of that. I wouldn't say we've locked it down yet. I'll show you the evidence we have. So, of course, the testis is famously the site for piRNA, the whole piRNA system of fighting off retrotransposons, or transposons generally. And so not only is the testis small RNA profile all piRNAs, but any purified sperm population you pull from the testis, in these pie charts, you can see green is the piRNAs, red is the tRNA fragments. This is true all the way out to mature testicular spermatozoa. Cauda sperm, which is what people use for in vitro fertilization and what I showed you data for before, is this red pie chart here, 80% TERFs. The caput epididymis has already completed this piRNA to TERF handover. Okay. That said, there are about 100 to 200 small RNA, but it's really as you leave the testis and enter the epididymis, you switch from piRNAs to tRNA fragments. Here you can see northern blots for a couple tRNAs. This is now looking at the epithelial cells of the epididymis. You see no tRNA cleavage in the testis, as I told you, and robust cleavage of tRNAs in the caput and cauda. So sperm leave the testis, go into a tissue that makes tRNA fragments, and the sperm gain tRNA fragments. The process by which we think that these RNAs are ending up in sperm is that one of the things that happens in the epididymis is that a wide variety of small vesicles, they'd be called exosomes elsewhere, here they're called epididymisomes, are known to fuse with sperm and deliver proteins to sperm. Exosomes are also famous for carrying small RNAs around. So it turns out it's quite easy to purify epididymisomes. When we clone small RNAs from them, they have more or less exactly the small RNA profile that sperm end up with. The exception that proves the rule here is that piRNAs are enriched in sperm, but absent from epididymisomes, which makes sense because the piRNAs were being carried forward from the testis. But all the tRNA fragments that sperm are going to gain are quite abundant in the epididymisomes. And then finally, we can recapitulate the delivery process ex vivo. So we can take testicular sperm, which are dominated by piRNAs, fuse them with epididymisomes, repurify the sperm, and so you can see that everything above this diagonal has been delivered by this fusion process. So we're pretty sure that the epididymis is synthesizing small RNAs to be delivered to sperm, to be delivered to the next generation. And this is sort of interesting because in a bunch of other gametogenesis systems, there's an element of soma to germline communication of small RNAs. And so we believe that the epididymis is the somatic tissue in mammals that delivers small RNAs to sperm. And so the image I want you to leave with is of the epididymis as sort of a novel sensory organ that responds to environmental conditions that the male experiences and modulates the epigenetic information that will be delivered to children. Okay. Now, the small RNA dynamics that occur in the male reproductive tract actually give us a really nice entree into thinking about the functions of small RNAs in the embryo. So I often ask myself, why aren't we doing this kind of work in flies or worms where we could do genetic screens pretty easily? And one thing that's kind of unique about mammals as a reproduction system is because of assisted reproductive technologies in humans, we have the ability to disassemble and reassemble the act of reproduction. What I mean by that is that you can take immature sperm from the testis, like round spermatids or testicular spermatozoa, and even though they can't swim and carry out IVF, you can inject those sperm into an oocyte to fertilize the oocyte. This is called intracytoplasmic sperm injection, or ICSI, and generate viable embryos. And in fact, human beings are made that way in certain cases of infertility. So we can take sperm from throughout the male reproductive tract, make embryos, look at their defects as offspring. We can look at molecular defects by a single embryo RNA-seq. And we can do all of that with or without fusing these sperm with the vesicles that will deliver the different stages of RNA. So in other words, you can take testicular sperm, which is all piRNAs, or cauda sperm, which is all TERFs, fertilize an embryo and ask what are the regulatory differences between the embryos made with these different sperm contributions. And so I'll tell you a little bit about our work using this kind of system. We actually have 4 different ways of manipulating the embryonic RNA payload. The obvious one is you can just make an embryo and then microinject a synthetic small RNA. We can also make embryos from sperm that are lacking entire classes of RNAs by germ cell specific deletion of PASHA for microRNAs. We're starting to do that kind of stuff now. What I'll tell you about is using the natural small RNA dynamics to ask about what happens if you fertilize an oocyte with sperm that carry all piRNAs versus all TERFs. And then lastly, you can make parthenotes. This is when you activate an oocyte to go through development without sperm at all. And then we can inject sperm total RNA and ask whether that has any regulatory functions on a blank slate in terms of sperm contributions. So I'll tell you briefly about the most developed system we've used here, which has turned out to be a little bit of a mess for us for unanticipated reasons. And so one of the first things we did is we looked at, so these sperm populations differ, they've already undergone the piRNA to TERF transition, but they differ in the expression of about 20 TERFs and 100 microRNAs. So we carry out ICSI. They have identical fertilization rates. They have identical blastocyst progression rates. These sperm are not grossly defective. At different stages of development, we carry out single embryo RNA-seq. This is very nice single cell RNA-seq data because, in a typical single cell RNA-seq data set, you'll get like 1 to 10,000 transcripts. We're clearly getting at least 1 to 10 million transcripts here, of course, because of the large size of the mammalian embryo relative to a fibroblast. So what we find is that when we use caput sperm to fertilize an oocyte, we overexpress about 50 genes. And it's a really interesting set of genes. They're all RNA regulatory proteins and chromatin factors. Here you can see dot plots for the individual embryo data. And here you can see that it's the misregulation profile is, pretty consistent or reproducible at multiple different stages of development. We also cannot make pups from these embryos. So when we implant these embryos into a pseudopregnant female, we get no pups out of our caput embryos. We do get pups out of cauda embryos. And this has turned out to be a mess because when we published the paper, we knew that other people had previously made caput offspring. And since we published the paper, we've had a lot of reproductive people point out that they can make caput offspring. We finally figured out, well, we didn't, somebody else has finally figured out what the discrepancy is, which is that we use an unusual way of making sperm heads. So the way other people make sperm heads is by sonication. We thought that that was a really violent way to break the sperm tail off, so we used thin needle shearing. But it turns out that if you use thin needle shearing, you get much lower pup production rates. So anyway, this has been kind of a mess to us, and it makes the next slide really confusing. Maybe I shouldn't even show it, but I haven't updated my slides in the last 6 months. Okay, so the question for us is what's wrong with caput sperm that leads to this misregulation and failure to implant? And so what we did is we made caput sperm ICSI embryos, and then after 3 hours after fertilization, so the zona pellucida hardens up and then softens again, we microinjected different populations of RNA. So we started out by taking cauda epididymysomes, gel purifying RNA and injecting it. And here's the overexpression of one of our target genes. Cauda RNAs suppress the misregulation we see with caput embryos. We then used size to separate microRNAs from TERFs. And you can see that the TERFs don't do anything, and it's cauda-specific microRNAs that rescue. We knew which microRNAs are differentially abundant between the caput and the cauda. We can synthesize these and rescue the caput gene regulation problem. And then when we go through the individual clusters, it turns out it's a really interesting cluster called the MER880 cluster. Here you can see the abundance in the testis, in the caput, and in the cauda. So it's 100-fold less abundant in caput sperm than in cauda or testis. It's an X-linked cluster. It's imprinted. It's a modifier of a hybrid incompatibility system, the PRDM9 system in mice. So it's just a really interesting and weird cluster, and it rescues all the gene expression problems with caput embryos. So this was a really beautiful result. However, what's really strange about it is that we now have to ask ourselves why it's only the case when you shear sperm through a needle as opposed to sonicating sperm that everything I just told you is true. So anyway, I apologize for just dumping a boatload of confusion in the middle of the talk. But this is being clear-eyed about the data. We have something that looks really interesting the way we perform the experiment, but there's something that we do that's weird that sort of reveals a sensitized need for this microRNA cluster. Anyway, I apologize. Let's move on to stuff that's a little bit less confusing. So anyway, this is something that we believe happens normally going through the epididymis, so this is not environmentally regulated. There's no difference in MER880 expression in low protein sperm or nicotine sperm or something like that. So let's go back to our diet regulated small RNA. I'm just going to skip that. This is a Parthenote experiment. Okay, RNA. So turf glycine GCC. And so we started out trying to understand the function of this tRNA fragment. So tRNA fragments are, generally speaking, an understudied set of regulatory molecules. For roughly a decade after they were first cloned, they were just assumed to be degradation products of really abundant RNAs, right? But people are increasingly coming around to the idea that these may have cellular regulatory functions. So what we did to start chasing these RNAs down is we just used an antisense inhibitor of TRFGG, and we started out in embryonic stem cells. So when we inhibit TRFGG, what we see is a de-repression of about 50 to 75 genes. They're de-repressed only when we knock down TRFGG, not when we use antisenses against other tRNAs, other isoacceptors, not when we use an antisense against the 3' of the tRNA. So we believe that it's the 5' TRF we're affecting, not the intact tRNA. All the genes that are overexpressed when we knock down our TRF had previously been described as having something in common, which is that all of them use as a regulatory element the long terminal repeat of an endogenous retrovirus called MERVL. So this was really exciting because, of course, we went down the tRNA fragment path because of the link between tRNAs and LTR elements. But it turns out that the way TRF glycine regulates the MERVL LTR has nothing to do with this ancient connection. And this is sort of obvious right away because the L in MERVL stands for I use leucine tRNA as a primer, and we're messing with glycine tRNA. The homology between tRNAs and the primer binding sequence is at the 3' end, and we have a 5' TRF, so on and so forth. So the MERVL regulon is quite famous. It was discovered by McFarlane and Pfaff and also Richard Schultz as basically the set of genes that are first turned on at embryonic genome activation. So in mice, you first wake up the embryonic genome in the 2-cell stage, and the first genes that turn on all use the MERVL LTR as their regulatory elements, whether it be promoters or enhancers. So you can see this here. By the time you get to a blastocyst, they don't express any of the MERVL program. What was really interesting is not only are these genes expressed in totipotent embryos, but in embryonic stem cells, the vast majority of cells in a culture are OCT4 positive. And if you take these cells and you aggregate them into an embryo, they contribute to the inner cell mass, but not to the trophectoderm, which means they contribute to the embryo per se, but not to extraembryonic tissues. This is why ES cells are pluripotent and not totipotent. What McFarlane showed is that if you sort out the rare MERVL positive cell, so about 2% of ES cells are in this 2C-like state. If you sort out these 2C-like cells and aggregate them into an embryo, they will contribute to the inner cell mass, but also to the trophectoderm. And so they'll contribute to the embryo, but also to extraembryonic tissues. And so our speculation was that because you see the same metabolic phenotypes in paternal effect paradigms as you see when you mess with the placenta, whether you starve a pregnant mom or tie off the uterine artery, what we speculated is that by regulating either how long you're totipotent for or how much of the totipotency program you express, we would basically be affecting cell allocation into inner cell mass versus trophectoderm and indirectly affecting nutrient acquisition via placentation. Okay, so we then tried to understand how our TRF is regulating the MERVL program. And so a postdoc, Anna Boscovich, pointed out that in general, zygotic genome activation is set by when you run out of histones. So for example, in the late ZGA species like Drosophila, you can actually or Xenopus, you can purify the anti-ZGA factor that prevents transcription, and it's the histone octamer. And we know that generally speaking, LTR elements are opportunistic about underassembled chromatin. So if you mutate chromatin assembly factor, the first transcriptional response is that LTR elements are de-repressed. So what Anna worked out is that what she believes is that TRF glycine GCC represses MERVL by supporting a supply of histone proteins. So it's very indirect. It has nothing to do with the fact that LTRs touch tRNAs in other contexts. And so the pathway that she sort of put together, I would say we're confident from here to here, less confident here. And so what she found is that when she knocks down our, when we transfect our TERF, we see higher histone levels. This is true of both the RNA and the protein level. Histones have a very unusual mechanism of regulation. Instead of using a polyadenylated 3' end, they have a little stem loop called the histone stem loop. It binds to a protein called SLBP. They also use a non-coding RNA that binds the histone downstream element. And what she found is that this non-coding RNA, U7, its levels do the same thing as the histone. So we knock down our TERF, we have lower levels. We transfect our TERF, we have higher levels. She also found that a bunch of other non-coding RNAs follow the same, basically the same behavior, which led her to speculate that our TERF is affecting the output of these phase separatist organelles called the Cajal bodies. Your colleague Carla Neugebauer, I don't know how to say her last name, studies these, of course. Okay, I'll give you one slide of evidence for this pathway. So when we knock down our favorite tRNA fragment, you see histones are downregulated. Here you can see by qPCR, repressing our TERF decreases histone levels, transfecting our TERF increases. We can domesticate this by putting the histone 3' UTR on luciferase. So again, downregulation and upregulation. The hint that this was going to operate through Cajal bodies came because in this data set, we also saw extreme downregulation of the SNO RNAs when we knock down our favorite TERF. You can see this here in CDF plots. And so here you can see U7, which is one of the other Cajal body outputs following the histones. And I think the killer experiment here is that we can rationally intervene with this process. So in other words, when we knock down our favorite TERF, we have less U7 and lower histone reporter activity. If we replace the U7 that we've lost, we can suppress the TERF knockdown effects on the reporter. So again, we believe what our TERF is doing is supporting non-coding RNA output of Cajal bodies, secondarily affecting histone levels, and thereby affecting zygotic genome activation. Okay? So this is sort of a cartoon of everything I just told you. Now, one thing that we've sort of been hesitant to do for a long time, and I can see I'm running out of time, so I may skip a little bit, but people always ask us, okay, so what happens if you microinject your tRNA fragment into a zygote? Does it have any phenotype? And so I was really hesitant to do that for a long time because the small RNA changes we see in low protein sperm included an upregulation of TERF glide GCC, but we also see a bunch of other small RNAs changing abundance, like we see lower levels of LET7. And I thought it was going to be really hard to sort of manipulate sperm RNA delivery so you add the amount of TERF glycine, but you halfway inhibit LET7, right? But so Anna finally just sort of had the balls to do the simple experiment, and she just injects TERF glycine GCC into control embryos, and she then transfers them to pseudopregnant females, and she gets a massive phenotype. And when I say massive, I mean literally. It turns out that the pups she's made after injecting TERF GG are about half again as heavy as control injected pups. So this is sort of preliminary. I think the N here is like 3 liters so far, but it's extremely exciting. And it's also challenging in a lot of ways. So first of all, this isn't the phenotype we see with low protein diets. Okay, we don't see massive pups. So it's not exactly like phenocopying what we see in the natural process. It also doesn't fit another experiment we have, which is I've been telling you about using ICSI to look at the effects of different RNA payloads. And if you'll recall, testicular sperm are all piRNAs and no TERFs. And yet when we carry out ICSI using testicular sperm, we don't get tiny pups, right? So they don't have any TERF glycine GCC, whereas cauda sperm do. So you would predict that the CAUDA fertilized embryos would end up big because they have the embigoning factor in them. But we don't see that. And, we know that this is the case because, again, humans are made using testicular ICSI at some, in some cases of infertility. So this is kind of a conundrum for us. I suspect what the answer is going to be is going to come back to the 3' ends of tRNAs. So as I mentioned, what we can clone easily from sperm are 5' TERFs. But we know from northern blots, okay, they're just very difficult to clone because the 3' sides of tRNAs have nucleotide modifications that interfere with reverse transcriptase. And so we actually have a collaborator now who has, and she doesn't want me to say who she is or how she does this, but she has a neat protocol for cloning all, full-length tRNAs or cloning 3' ends. And sure enough, sperm are filled with these 3' ends. And so now what we're sort of returning to is what actually are sperm delivering to the embryo. So we know they're delivering a 5' TERF, which we know in isolation affects the histones and affects MERVL and affects the size of the pup. But now when you also deliver the 5' and the 3' end, like is perhaps the difference between CAUDA embryos and testicular, even though CAUDA have the 5' ends, they also have the 3' end. And so maybe what's going on is that, effectively a nicked tRNA that's complexed is going to interfere with the activity of the 5' TERF. And so Anna's done one experiment where she injects embryos with an annealed 5' and 3' TERF, and sure enough, they don't show this size increase. And so now we think that probably what we ought to be thinking about is less what is the level of 5' TERF glycine in sperm, but rather what's the level of free 5' TERF glycine. So what's the ratio of 5' to 3'? And we're trying to figure out ways of actually directly assaying free versus bound up tRNA fragments. Okay. I guess I'll leave you with one other thing and then a question, and then I'll skip the nicotine stuff. The other issue with the other curious aspect of our phenotype when we inject TERF glycine is that this massive pup phenotype has also been seen by Minu Russell-Zadigan, I do know how to say her name, when she injects miR-124 into embryos. So they also get massive pups. And so this sort of raises, again, a question that we see raised in a bunch of different areas of paternal effect studies. So how do TERF glycine and miR-124 give you similar phenotypic outcomes? Another way of thinking about this is if we think that what our TERF is doing is accelerating or decelerating zygotic genome activation, how does that tell you anything about a low protein diet? If dad wakes you up early, it could be that the house is on fire, or it could be that it's Christmas. And so the $1 million question for us in the paternal effect literature, I think it's now more or less indisputable that a father's world affects phenotypes in kids, but how much information can dad pass on to kids? In other words, what is the bandwidth of sperm? So if you think about cytosine methylation, you have 25 million CpGs, each can be a bit of information. You could tell your kids extremely high resolution information about the world you saw. Cadmium levels, zinc levels, I like the color red, mates are abundant in the fall, whatever. On the other hand, there's a bunch of reason to think that maybe all you tell your kids about is some overall quality of life metric, right? Like life was shitty, okay, or great. And there are a bunch of reasons to expect this kind of model, right? For example, while the people who study paternal diets see metabolic phenotypes in kids, people who study paternal stress see stress phenotypes in kids. When the stress people look for metabolic phenotypes, they find them. And so it may be the case that all of these different things that we do to dads in the end are just affecting an intergenerational stress response. So you just tell your kids overall quality of life. Alternatively, you could imagine something, somewhere in between these extremes, where maybe you tell your kids about like 3 or 4 dimensions, salient dimensions of the environment, right? Like GlyGCC is nutritional stress, GLU-CTC is psychic stress, and VALCAC is water. And so this is now something we're very keen to try to understand. It's for this reason that we developed a nicotine system, because while low protein diet does an infinite number of things to an animal, nicotine binds to nicotinic acetylcholine receptors, which means that we can use the tools of pharmacology to assess the specificity of the offspring phenotype. Anyway, I'll skip that. It's published, Vallister et al. In eLife. The implication of the nicotine system seems to be that you tell your kids extremely low specificity information. So kids, briefly, if dads are on nicotine, the kids are resistant to a lethal dose of nicotine, but they're also resistant to a lethal. So in other words, you're not downregulating nicotinic receptor alpha 4, you seem to be making your kids xenobiotic resistant. And the signaling in dads doesn't require nicotinic acetylcholine receptor signaling. And so we really quickly go from nicotine affects nicotine responses in kids to small molecules in dads affect small molecules in kids. So in other words, we, at least from the nicotine system, we very much favor a low bandwidth hypothesis. So sorry that I had to race through bits and pieces of it. I think what I'd like people to take away from this is that hopefully I've convinced you that there is a huge amount of literature that now confirms that a father's environmental exposures do modulate physiology in the next generation. I do not believe that we understand how it works, although I do believe that when you inject RNAs into zygotes, you can change phenotypes in kids. I'm pretty sure that the RNAs that sperm deliver to kids are actually first made in the somatic cells of this tube called the epididymis. Certainly the sperm RNA payload changes dramatically as it goes through the epididymis. We like to think of it as a sensory organ that modulates the RNAs that sperm deliver to kids. And then finally, at least with our favorite small RNA, turf glycine GCC, we think that what it does is it regulates how early, how quickly, or how much you turn on the zygotic genome. And we speculate that in the end, that affects metabolic phenotypes by affecting placentation. So this was the lab at its peak, although, you know, these guys weren't working in the lab, they're just the F1s. All the work I told you about was really driven, all of the stuff in the epididymis was driven by Upasana Sharma, who did all of the epididymisome work and started the turf glycine work. She just started her lab at UC Santa Cruz. Colin Conine was our small RNA expert. He did all of the ICSI work I told you about, just started his lab at Penn. All of the mechanistic work on our turf was done by Anna Boskovich and student Shenyang Bing. Nicotine system was carried out in collaboration with a great colleague, Andrew Tapper. And we've leaned on our small RNA colleagues, especially Zamor and Mello, over the years to try to keep our heads above water. And thank you for showing up, and I'll take any questions.
All right, thanks. So I guess I can, if you want to use the raise hand function, I'll look out for that. And otherwise, I see there are already some questions in the chat.
Shall I start with the chat questions?
Sure, although I'm going to jump in and take advantage of my hosting status first. So I thought the idea of this balance between the 3' and 5' fragments is really cool. I guess I wasn't quite clear on what the link is. So do you think in the paternal diet models that there's an imbalance between the 5' and 3'?
So we don't know. So but that's, I mean, that is now for me the $1 million question. So, you know, when we see an increase in the 5' turf, is it that there's an increase, a concomitant increase in the 3' turf, which would still lead to, you know, KDs being the way they are with a small increase in the amount of free 5'? Or is there sort of, as you say, an imbalance where you see an increase in the 5' turf because you're cleaving and, you know, degrading more of the 3'? You know, the problem we're having is that even with this new protocol, where we can see 5' and 3' turfs, we don't have any great way of benchmarking the extent to which we're quantitatively capturing the 3' or the 5'. And it's while we can like just take a synthetic 5' and 3' and mix them in precisely equal molar ratios and try different cloning protocols on them, that's actually not as easy as it sounds because of the nucleotide modifications you need to put onto them. And many of these are actually not phosphoramidites to make them synthetically. So it's actually a much more challenging problem to benchmark than you would think. What we've been trying to play with is figuring out some way of like trying to cross-link an annealed nicked tRNA, like with something like sorrelin, and then running a northern blot and seeing how much of the 5' side shows up running free versus cross-linked. And we're trying to do that with a postdoc who came out of Sean Ryder's lab. So we have, who's a Yale alumnus. Anyway, so we're thinking about that. I think the number of nucleotides or base pairs of stem you need for sorrelin might be a little bit longer than you're going to get in the acceptor and acceptor and anticodon stems of a tRNA. But anyway, so your question is a great one that we are expending a lot of thinking but very little functional experiments on. But I think it is the $1 million question, because if Anna's current result holds, which is that injecting the 5' alone changes the size of pups, but annealing the two and injecting the same amount of 5' but now interfered with by equivalent 3' doesn't change the size, then that means that the absolute amounts of 5' you clone mean nothing, right? It means you really have to understand the active free 5' side.
All right, thanks.
Okay, shall I start with June now?
Sure, yeah.
June Liu, how many molecules of TERF are in a single sperm? Is the level sufficient to cause changes in histone levels? That's a really good question. We basically have the same problem with that question that we have with, so I'll tell you a little bit more about the new cloning protocol we're using. So with the 5', when we clone with TrueSeq, tRNA fragments are all 5' and they're like 80% of all small RNAs we clone. With this new protocol, which uses one of these target-directed, it's like a line 1 reverse transcriptase that can sort of jump between small RNAs, we see similar levels of 5' and 3' TERF, but now TERFs are much less relatively abundant. We're still trying to figure out where all the other reads are coming from, because they don't come from well-known small RNA classes like microRNAs. So at this point, we just don't even know what the ground truth is of what RNA molecules sperm are carrying. We've done spike-ins to attempt absolute quantitation, and with TrueSeq, the different RNAs we spike in don't follow the ratios we spike them in at. In other words, if you do 10-fold, 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 10-fold for 4 spike-ins, these two will differ by 10-fold, and then these two, which should be 100 and 1,000-fold lower, will be at these two levels. So TrueSeq is a disaster in terms of absolute quantitation. I think it's super sensitive to whether or not the 3' nucleotide is a G, this kind of stuff. So I don't believe we know the absolute quantitation. That said, our best estimates have been on the order of 100,000 RNAs, small RNAs per sperm. But basically, I'm going to tell you that I do not believe that number at all. I don't even believe we know what sperm are carrying anymore. And again, it's just going to be really hard to figure out the correct spike-ins to do the experiment, because you can't really make synthetic tRNA fragments with the right modifications to even ask the question. So sorry, it's a bit of a punt. What I can tell you in terms of the level question is when we inject a synthetic tRNA fragment, we are attempting to inject the number of molecules that we are estimating are in there. So with TERF glycine GCC, on the order of 5 to 10,000 molecules. That does affect MERVL in the injected zygotes. So, with the synthetic molecule, we can say that we're injecting to plus minus 2 to 3-fold, 5 to 10,000 molecules, and that is sufficient to cause MERVL changes. So I think it is enough, but we don't really know what's actually in there. Okay, Antonio. Hi, Antonio. I have 2 questions. Do you see genes that are not misregulated that are also controlled by MERVL when you misregulate TRFGG? I'll answer that first. So yes, so the McFarlane MERVL program is about 300 genes, which were defined in a bunch of ways. One of them was having a chimeric transcript from the LTR through the gene. We misregulate 50 to 75. So Anna calls these the core MERVL program. So of the famous MERVL program, so we are misregulating the TD pause genes, SP110, the DUBs, the USPs. We are not misregulating the Z scans, which is the other big famous MERVL family. What I don't know the answer to is if you like knock down CAF1 and you de-repress the MERVL program, is our subset the same subset that would be misregulated by CAF1? And that's something we should do. But so I don't know the answer to the question of whether or not our 50 to 100 are the particularly histone dosage sensitive MERVLs. Question 2, from the zygotic genes expressed at ZGA, what fraction of them are affected? So TRFGG, we only affect 50 to 100 genes. And of course, if you take the whole ZGA program, it's several thousand genes. Now, one thing that you can do when you have the kinetic resolution that ICSI gives you, so most of the time when you make an embryo, you have a slop factor of about 4 hours, even through IVF, right? But with ICSI, you know the moment of fertilization to within 5 seconds. We made, a bit of a time course between 18 and 28 hours. And so there's like early and late ZGA, even within the major ZGA wave. And of course, the major is the MERVL program. I wouldn't care to tell you that that's 500 genes or 200 genes, but it's not all 5,000. So I think we're still affecting a subclass of the early ZGA, but it's, a higher fraction than if you included the whole like 5,000 gene ZGA. Okay, Cindy Barish. Do you think the small RNAs are maintained over time in the F1s? So this is one of the really interesting things about doing this in mammals. So the species where people can do like genetics of epigenetics, right? Like RNAi in worms or paramutation, those species all have encode an enzyme called RNA-dependent RNA polymerase. And so when you increase the levels of a small RNA, you can maintain the increased levels of that small RNA by copying. Mammals don't encode an RDRP, and so anything that's small, the reason we do all of this like mucking around with preimplantation embryos is that if sperm delivers a small RNA, the levels, at least as we understand the molecular biology of RNAs in mammals, you will not be maintaining higher levels of that small RNA for more than a cleavage or 2, because they'll be diluted out. That's not to say it's implausible, right? So with you could imagine that a microRNA or a piRNA directs a histone modification or something to the locus that encodes it, and then affects transcription in perpetuity and maintains high levels of that RNA. With TERFs, it's a little bit harder to think about, right? Because even directing a histone modification to a tRNA locus might not necessarily affect cleavage. So I'm just going to say that generally speaking, the trick in mammals is that we don't believe that small RNAs delivered to the zygote are maintained for any long period of time. So you ring the bell at fertilization, and then everything that happens later is secondary, tertiary, quaternary consequences. And then the second part of your question, once they reach their lifespan, how does the offspring know to maintain them? And as I just said, I believe that the offspring does not maintain small RNA levels following fertilization. Okay, back to June. Why a change in histone levels would have such a specific effect on a small number of MERVL regulated genes. Well, there are two ways to answer that question. In terms of why it has a specific effect on LTRs, it's the case. So if you delete CAF1 in yeast, basically the only transcriptional consequence is that you de-repress TY elements. It's simply the case that LTR elements are, have evolved to be extremely opportunistic about underassembled chromatin. So that's why it's specific to an LTR element, is they're the ones who take off the second the histones let up. Why it's a subset, this is sort of like Antonio's question, why is it the TD pauses and not the Z scans? I don't know the answer to that. And again, as I answered his question, we haven't looked at, like, in a CAF1 mutant, are the TRFGG targets specifically more sensitive to underdosing with histones? So anyway, we don't know, but that would be the obvious speculation.
All right. Are there any more questions? Last minute. Okay. Well, thanks, Ollie. And I guess I'll stick around for a minute here. I know you have to go in a minute too. And see everyone else later.
Thank you all for coming. Antonio, see you later.
Thank you, Oliver. I'll see you later.